Hello, sir. I'll proceed, sir.
Yeah, please proceed, madam.
Okay, sir. Hello, everyone. Welcome to Tawhid Foundation for Allied Healthcare Sciences, Bangalore. Today's class is about Immunology Part 3, Antigen Antibody Reaction for BSc MLT third year students. So today we are going to learn about one of the most important concept in immunology, that is the antigen and antibody reaction. This is the basic reaction that allows our immune system to recognize and defend against anything foreign, whether it is a virus, a bacterium, or a toxin. Whenever a specific antibody encounter its matching antigen, they fit together like a lock and key, forming an antigen antibody complex. So this simple interaction is not only what protects us from disease, but it also forms the foundation of many diagnostic tests used in laboratories such as ELISA, agglutination, precipitation, and complement fixation. So by understanding how these complexes and reactions form and behave, we can better understand how our immune system works and how modern medicine detects and prevents infections. So let's proceed. The antigen antibody reaction is the fundamental event in immunology where a specific antigen binds to its corresponding antibody. This interaction is the very basis of immune recognition and defense in our body. Every antigen has certain unique parts called antigenic determinants or epitopes, and antibodies have specific region known as paratopes. The binding occurs because these two structures are complementary in shape and charged like a lock and key, allowing them to fit together precisely. The specific binding forms the foundation for almost all these tests in clinical immunology, such as ELISA, agglutination, precipitation, and radioimmunoassay. In simple terms, whenever an antibody meets its exact antigen, they combine to form antigen antibody reaction, which can then trigger visible or measurable reaction used in laboratory testing. This reaction is the reason our body can recognize and neutralize harmful pathogens accurately. Now, what is the role in immunity and diagnosis? Antigen antibody reaction plays a crucial role in both immune protection and medical diagnosis. In the immune system, these reactions act as the main defense mechanism against invading microorganisms. When antibodies recognize antigen on the surface of pathogen, they either neutralize them directly or mark them for destruction by immune cells. For example, antibodies can block toxin released by bacteria or prevent viruses from entering the host cell, a process known as neutralization. In diagnostics, the same principle is applied to detect the presence of either antigens or antibodies in patient samples. Tests like ELISA, Western blot, or agglutination are based on observing this specific reaction. These tests help in the detection, quantification, and identification of diseases by measuring how strongly an antigen and antibody interact. So this reaction not only protects us but also helps doctor identify infections accurately. Historical background. The study of antigen antibody reaction has a long and fascinating history. In the 1890s, Emil von Behring and Shibasaburo Kitaso Sato discovered that serum from animals immunized against diphtheria contained substances that could neutralize the toxin. This discovery gave birth to the concept of serum therapy and laid the foundation of immunology. Later, Karl Landsteiner's work revealed that antibodies were specific, meaning once antibody reacts with only one particular antigen. He also discovered blood groups and explained how antigenic determinants control specificity. So, in 1934, Marek proposed the lattice hypothesis, explaining that precipitation and agglutination occur when multivalent antigens and antibodies form large interconnected lattice. As technology advanced, newer immunological tests were developed, such as radioimmunology assay in the 1960s, ELISA and Western blot in later decades, which greatly improved the sensitivity and accuracy of detecting antigen antibody reactions. So, together, these discoveries transformed immunology into one of the most precise and diagnostic sciences in medicine. Next is importance in medicine. Antigen antibody reactions are the backbone of modern medicine and diagnostic science. They are essential in vaccine development. Vaccines work by stimulating the body to produce antibodies against a specific antigen, providing long term protection. In laboratories, these reactions help in the diagnosis of various infections and autoimmune diseases. For instance, detecting antibodies against HIV or hepatitis B antigen in a patient's serum helps confirm infection. These reactions also assist in monitoring the spread of diseases in populations through serological surveys, which detect who has been exposed to a pathogen. Beyond diagnosis, antigen antibody technology has immense value in biotechnology, microbiology, and research. It is used for developing therapeutic monoclonal antibodies, designing diagnostic kits, and studying immune disorders. Thus, this reaction connects immunity, diagnosis, prevention, and treatment together in clinical practices. Stages of reaction. The antigen antibody reaction occurs in three main stages: the primary stage, the secondary stage, and tertiary stage. In the primary stage, the antigen and antibody first recognize and bind to each other. This is a microscopic process and cannot be seen visually. It involves the formation of an antigen antibody complex. Secondary stage, these complexes become visible through reactions like precipitation or agglutination, depending on the nature and concentration of the reactant. Tertiary stage includes the biological effects produced in the host due to this reaction, such as complement activation, cell destruction, or immune complex diseases. So each of these stages has its own importance. The primary stage shows molecular recognition, the secondary stage is used in laboratory tests, and the tertiary stage explains the immune response seen in living systems. We'll go one by one. Primary stage and its features. The primary stage is the initial and invisible part of the antigen antibody reaction. Here, the antigen and antibody come close enough to form a non covalent bond like hydrogen bond, electrostatic bond, and hydrophobic interactions. These forces allow reversible binding between the two molecules. This binding is extremely specific. The paratope of an antibody fits exactly to the antigen's epitope, much like a lock fits a key. Even a slight change in the structure of the antigen can prevent binding. Since this stage does not produce any visible change, it can only be detected by sensitive laboratory techniques like ELISA or radioimmunoassay. The strength of binding depends on the affinity of the antibody and the number of binding sites on both antigen and antibody molecules. These microscopic binding forms the foundation for all visible reactions that occur in later stages. Next is secondary stage. In the secondary stage, the antigen antibody complexes that form during the primary stage become visible. When multiple antigen molecules and antibodies cross-link, they form large network or lattices that can either precipitate in solution or agglutinate cells. The visibility of this reaction depends on the concentration and valency of the reactants. Both antigen and antibody must be multivalent to form a lattice. An important concept here is the zone phenomenon, which describes how reaction outcomes vary depending on antigen and antibody concentration. In the prozone, that is antibody excess, no visible reaction occurs because every antigen is covered by antibodies. In the equivalence zone, optimal proportions lead to maximum lattice formation and visible precipitation. In the post zone, that is antigen excess, small complexes form but remain invisible. Understanding these zones helps in accurate laboratory diagnosis and prevents false negative results during tests like precipitation or agglutination. So, it is useful in laboratory diagnosis. Next is tertiary stage. The tertiary stage represent the biological consequences of antigen antibody interactions occurring inside the body. Once the antigen and antibody combine, they can activate several immune mechanisms. One such mechanism is complement activation, where a series of serum proteins are triggered to destroy foreign cells by lysis. Another is opsonization, where antibodies coat antigens or bacteria, making them easier for phagocytic cells to engulf. These reactions provide immune protection against infections by neutralizing toxins and pathogens. However, not all tertiary reactions are beneficial. Sometimes the immune complexes deposit in tissues and cause inflammation or hypersensitivity. Hypersensitivity reactions such as autoimmune diseases like lupus. Therefore, tertiary stage outcomes determine whether the immune response helps the body fight disease or causes tissue damages. Next is antibody titer concept. Antibody titer is a measure of the concentration or strength of antibodies present in a patient's serum. It is determined by a process called serial dilution, where the serum is progressively diluted and tested until the antibody activity is no longer detectable. The highest dilution that still shows a visible or measurable reaction is considered the titer. For example, if the reaction is visible up to dilution of 1 is to 64, the antibody titer is reported as 64. A high antibody titer indicates a strong immune response or recent exposure to an antigen, while a low titer suggests either recovery or absence of infection. In medical diagnosis, comparing antibody titer in two serum samples taken at different times helps determine whether an infection is recent. A fourfold rise in titer usually confirms an active infection or successful immunization. Next is antigen titration. Antigen titration is the process of determining the amount of concentration of antigen present in a sample. It is performed by using a known amount of antibody and measuring the point where the antigen antibody reaction gives an optimum visible result. This process helps in standardizing diagnostic tests and ensuring that reagents have accurate and reproducible concentration. For example, in ELISA or immunodeficiency, antigen titration helps identify the correct dilution that produces measurable results without excess or deficiency. Accurate antigen titration is especially important for quality control in diagnostic kit manufacturing, vaccine production, and experimental immunology. It ensures that test results are reliable, comparable, and consistent between different laboratories and batches of reagent. Next is sensitivity and specificity in test. Sensitivity and specificity are two fundamental characteristics of any diagnostic test. Sensitivity refers to the test ability to detect even small quantities of antigen or antibody, meaning it can identify almost all true positive cases A highly sensitive test reduces the chances of missing an infected person, hence preventing false negatives. Specificity, on the other hand, is the test ability to react only with the intended antigen or antibody, thereby avoiding false positives. In medical testings, high sensitivity is preferred for screening tests where the goal is to detect all possible cases. High specificity is essential for confirming tests to ensure that the result truly represents the disease in question. Therefore, an ideal diagnostic test maintains a balance between sensitivity and specificity, ensuring that patients receive both accurate and reliable results. Next is definition and requirements. Precipitation is a type of antigen antibody reaction that occurs when a soluble antigen reacts with its specific antibody in optimal proportions to form a visible insoluble precipitate. So for this reaction to occur, the antibody must be bivalent, meaning it has at least two antigen antibody sites, and the antigen must be multivalent, containing multiple repeating epitopes. So the interaction between these two allows the formation of large cross-linked complexes that settle out of solution as a precipitate. This reaction only happens when both antigen and antibody are present in the right proportions. Too much of either will prevent lattice formation. Precipitation reaction form the scientific foundation of immunodiffusion techniques, which are widely used for both qualitative and quantitative detection of antigens and antibodies in laboratory diagnostics. Next is lattice hypothesis concept. The lattice hypothesis proposed by Marek in 1934 explains the basic mechanism of how antigen antibody complexes form during precipitation reactions. According to this theory, precipitation occurs when antigen and antibody molecules combine in such a way that they form large cross-linked networks or lattices. These lattices are insoluble and can be seen as visible precipitates in a test tube or gel medium. The formation of such lattices requires that both the antigen and antibody molecules possesses multiple binding sites. A single antigen molecule must bind to more than one antibody molecule and vice versa. If either is present in excess, large lattice structures cannot form and no visible precipitation will be observed. Thus, the lattice hypothesis provides a scientific explanation for why optimal antigen antibody ratios are crucial for visible reactions. Next is zone phenomenon. The zone phenomenon describes how the relative concentration of antigen and antibody affects precipitation. There are three zones. First is prozone, equivalence, and post zones. In the prozone, there is an excess of antibody which prevents the proper cross-linking of antigen molecules, resulting in no visible precipitation even though antigen and antibody are both present. In the equivalence zone, the antigen and antibody are present in optimal proportions, allowing maximum lattice formation and producing the greatest amount of visible precipitate. In the post zone, there is excess antigen, which also prevents lattice formation since most antibody binding sites become saturated by single antigen molecules. Understanding these phenomenon is very important in clinical testing because inappropriate concentration of reactants can give false negative results, misleading diagnosis. Therefore, proper dilution of sera is always essential for accurate immunological testing. Next is a precipitation curve diagram, this schematic representation. So First, let's look at this diagram. It is called the precipitation curve or antigen antibody ratio curve. These graphs explain the relationship between the relative concentration of antigen and antibody and the amount of precipitate formed during a precipitation reaction. So understanding the axis, on the x-axis, that is horizontal line, we have the increasing ratio of antigen to antibody, meaning as we move from left to right, the amount of antigen increases while antibody stays constant. On the y-axis, that is vertical line, we have the amount of precipitate formed. That's the measurable outcome of the reaction. Now look at the shape of the curve. It rises, reaches a peak, and then it falls. And then it falls. These represent three distinct zones in an antigen antibody reaction. So we'll see these three zones one by one. A, that is zone of antibody excess, this left side of the curve. At the far left, where antibody concentration is very high and antigen concentration is low, we have the zone of antibody excess. Here, each antigen molecule is surrounded by antibody molecules because there are too many antibodies and not enough antigen molecules to cross-link them. Large lattices cannot form and therefore very little or no visible precipitate is produced. So in this region, antibody is excess, small soluble immune complexes form, or little or no precipitation is seen. Next is zone of equivalence, that is middle peak of the curve. As we move toward the center of the curve, the ratio of antigen to antibody becomes optimal. This is the zone of equivalence. In this zone, the number of antigenic sites and antibody binding sites are approximately equal. This allows maximum cross-linking between antigen and antibodies, forming large insoluble lattice networks. Because of this extensive lattice formation, maximum precipitation occurs here and the curve reaches its highest point. This is the ideal region for performing serological tests because reactions are clear and visible. Next is zone of antigen excess, that is right side of the curve. Now as we move further to the right, antigen concentration becomes much higher than the antibody concentration. This is the zone of antigen excess. Here each antibody molecule becomes saturated with antigen and there aren't enough antibody molecules to link the antigen particles together. As a result, only small soluble complexes form and the amount of visible precipitate decreases again. The curve slopes downward. So in this region, antigen is in excess, no lattice formation occurs, precipitation decreases. So here on the left, too much antibody, not enough antigen, that this antigen is suckle, so not enough antigen, so no cross-linking. Here in the middle, perfect balance, large lattice form, maximum precipitate, and here on the right, too much antigen, antibodies are saturated, no more lattice formation, so precipitation falls again. So this figure is extremely important because it explains why immunological tests such as precipitation, immunodiffusion, or immunoelectrophoresis only give clear visible results when antigen and antibody are present in optimal proportions. If either one is in excess, you won't get a visible reaction even though both are present. This concept is known as the prozone and postzone effect. So the maximum amount of precipitate formed at the zone of equivalence, where antigen and antibody concentration are roughly equal, too much of either antigen or antibody prevents proper lattice formation and reduces precipitation. These principles form the basis of all quantitative precipitation test in immunology. Next is precipitation in gel. The precipitin ring test is a simple tube method used to detect an antigen antibody reaction in solution. It demonstrates how soluble antigen and antibody interact to form a visible ring of precipitate at their zone of equivalence. So let's first look at this figure A, which shows a schematic drawing of the test setup inside a small narrow test tubes. At the start of the test, we carefully layer antigen solution and antiserum, that is antibody, one above the other in the tube. Usually, antiserum, that is antibody, is placed at the is placed first at the bottom of the tube, as you can see here. Then antigen solution is gently layered on top, so the two liquids form distinct layers without immediately mixing. Now, over time, both antigen and antibody molecules begin to diffuse slowly towards each other through the liquid interference. The antigen molecule diffuse downward and the antimolecules diffuse upward. This antigen molecule will diffuse downward and antibody will diffuse upward. At a certain point between the two layers, their concentration become exactly equal. This region is called the zone of equivalence. Here, the antigen antibody complexes form large insoluble lattices that appear as a visible white ring or band at the interference between the two layers. So, that's the precipitin ring, marking where antigen antibody met in optimal proportion. If the antigen concentration is high, the ring forms lower, that is closer to the antibody layer. And if the antibody concentration is high, the ring forms higher, that is closer to the antigen layer. So the position of the ring can give a rough idea of the relative concentration of antigen and antibody. This is the actual photograph. Now, this figure B, which is a photograph of the real result, here you can see a clear white ring or opaque band in the middle portion of the tube. That ring is the precipitin line formed by the antigen antibody complex at the zone of equivalence. The upper liquid layer represents the antigen solution and the lower layer is the antibody containing serum. The sharp visible ring in between is the proof of antigen antibody interaction. So, the test works on the same principle as all precipitation reaction. When soluble antigen and antibody react at optimal concentrations, they form an insoluble complex that become visible as a precipitate. The reaction occurs in a liquid medium, so the precipitate collects as a ring where the diffusion fronts meet. So here the antigen layer on top in the and antibody layer below. So as they diffuse toward each other, they meet at the zone of equivalence and form the visible precipitin ring. So the precipitin ring test visually demonstrate the basic concept of antigen antibody precipitation in a liquid medium. When antigen and antibody diffuse towards each other and meet in equivalent proportion, they form a visible white ring of precipitate at their junction, conforming a positive reaction. Next is clinical importance of precipitation. Precipitation reactions are extremely important in clinical microbiology and immunology. They are used for the detection of fungal antigens, such as in the diagnosis of Aspergillus or Candida infections. They are also useful for identifying bacterial toxins, for instance, in diphtheria and anthrax, by detecting specific antigens in patient samples or culture extracts. In vaccine production, precipitation techniques are used for quality control, ensuring that the right amount of antigen is present and that the vaccine components are immunologically active. Additionally, precipitation reaction are the basis of many immunodiffusion tests, which allows for the detection and quantification of antigen antibody interactions in gel media. These applications show how a simple reaction can be effective diagnostic and research tool in medicine. Next is precipitation in liquid ring test. The ring test is one of the simplest precipitation reaction performed in liquid medium. In this method, a layer of antigen solution is carefully added over a layer of antibody solution in a narrow test tube, as I mentioned in this schematic representation of precipitation in gel ring test. So, at the interference where the two solutions meet, antigen and antibody diffuse slightly into each other and react at the zone of equivalence, forming a visible ring of precipitate. This ring confirms the presence of specific antigen or antibody in the sample. The test is rapid and easy to perform, requiring no special equipment. A classical example of the ring test is Ascoli's thermoprecipitant test, which is used for the identification of anthrax antigen in infected animal tissues. Because of its simplicity and specificity, the ring test remains a useful screening tool in diagnostic laboratories. Next is precipitation in liquid flocculation and tube test. In flocculation test, fine particles, that is fine antigen antibody complexes, form and appear as visible clumps or flakes rather than a dense ring or pellet. These aggregates result from the combination of soluble antigens with antibodies under conditions that favor partial precipitation. One of the most well-known flocculation tests is the VDRL test, which is used for the diagnosis of syphilis by detecting antibodies to cardiolipin antigen. Flocculation reactions are sensitive and can detect even low concentration of antibodies. In the tube precipitation test, both antigen and antibody are mixed in varying concentration in test tubes, and the resulting precipitate is allowed to settle at the bottom. The intensity or amount of precipitate can be measured to quantify antigen antibody concentration. Both these liquid phase precipitation methods are widely used in clinical diagnosis and serological standardization. Next is precipitation in gel general. So in gel precipitation, also known as immunodiffusion, both antigen and antibody diffuse through a semi-solid medium such as agar or agarose. As they move through the gel, they eventually meet at a point where the concentration reaches the zone of equivalence and a visible precipitate line forms. This line remains stable and can be easily visualized, making the method reliable for qualitative and semi-qualitative analysis. An important advantages of gel precipitation is that it allows multiple antigen antibody systems to be tested simultaneously on the same gel plate. This technique is simple, does not require complex instruments, and can reveal a relationship between different antigen and antibodies based on the pattern of precipitin lines. Thus, precipitation in gel is an essential method in immunodiagnostics and research. Immunodiffusion precipitin gel reaction in gel. So now we are looking at one of the most important techniques in immunology, the immunodiffusion test, which is a modification of the simple precipitin reaction. Instead of allowing antigen and antibody to react in a liquid medium like the ring test, here both are placed in small wells cut into an agar or agarose gel, and they diffuse through the gel towards each other. Where they meet at optimal concentration, they form a visible precipitin line, also called as precipitin band, as you can see in this diagram. So look at part A of the figure, this shows a diagrammatic representation of the setup. You can see an agar plate represented as a flat rectangular block. Two small circular wells are cut into the gel. One well is filled with antigen, and that is antigen solution. The other well is filled with antibody solution. Once the well are filled, both the antigen and antibody molecules begin to diffuse outwards slowly through the gel in all directions. The arrows in the figure shows the bidirectional diffusion, that is the antigen spreading from its well and the antibody from its own. As they move through the gel, their concentration gradually decreases with the distance from the well. At a certain point between the two wells, the concentration of antigen and antibody becomes equal. This is called the zone of equivalence. At this point, the antigen and antibody interact and form an insoluble antigen-antibody complex. These complexes become trapped in the gel, appearing as thin white opaque line, which we call a precipitin band. So as you can see, that's the visible evidence of antigen-antibody reaction. Now look at this figure B, which shows the real appearance of the precipitin line after staining or visualization. You can see two wells in the gel, one containing antigen and another one containing antibody, and between them a clear white curved line or band has formed. That band is the precipitin line, marking the zone where the antigen and antibody diffused together in equal proportions and formed an insoluble complex. So this line remains stable in the gel and can be visualized directly or stained to make it more prominent or more visible. Principle, the immunodiffusion technique used or works on the principle that both antigen and antibody are soluble and can diffuse through a semi-solid gel. When they meet in optimal proportions, they form an insoluble complex that remains as a visible line in the gel. The line forms at the exact position where their concentration are equivalent. Importance of this precipitin band. The position of the precipitin line depend on the relative concentration of both antigen and antibody. If the antigen concentration is higher, the line forms closer to the antibody well. If the antibody concentration is higher, the line forms closer to the antigen well. So by observing the position and number of lines, we can analyze the specificity and relationship between different antigens and antibodies. So one in whole, immunodiffusion is a gel-based precipitation test in which antigen and antibody diffuse through agar towards each other and form a visible precipitin line where they meet in optimal proportions. This line is called precipitin band, indicate a successful antigen and antibody reaction. Next is immunodiffusion. So immunodiffusion, as I mentioned earlier, it is a serological technique in which both antigen and antibody diffuse through a semi-solid medium, usually agar gel. When they encounter each other in optimal proportion, they form a visible precipitin line representing antigen-antibody complex. This reaction can be carried out in one or two dimensions depending on the setup and diagnostic purpose. Immunodiffusion can be used for qualitative detection, such as identifying whether an antigen or antibody is present, and for quantitative estimation, such as measuring the concentration of immunoglobulins or specific proteins. Because of its accuracy, simplicity, and stability, immunodiffusion remains one of the most commonly used serological methods in diagnostic immunology. Next is single diffusion in one dimension, Audin procedure. The Audin procedure is the simplest form of immunodiffusion performed in one dimension. It is carried out in a test tube or capillary tube where a column of agar gel contains a uniformly distributed antibody. A layer of antigen solution is carefully placed on top of the agar. The antigen molecules slowly diffuse downward through the gel and meet the antibody molecules within it. At the point where their concentration reach equivalence, a single sharp precipitin line appears. This line indicates the antigen antibody reaction zone and confirms the specificity of the antibody for that antigen. The Auden test is used primarily for qualitative identification of antigen and is the foundation for more advanced diffusion techniques. So in this setup, you can see a rectangular gel block or stripes. Inside the gel, the antibody is uniformly distributed throughout the entire gel, act as a reservoir of antibody. On the upper surface or at one of the end of the gel, there's a narrow